Hello everyone, welcome back. Here in this presentation we will be looking into one of the biologically important nucleotide. Now before that we have to understand what are the biologically important nucleotides. We have already seen the nucleotides, how it is formed, nucleic acid, the structure of different types of nucleic acids, etc. But which all are the biologically important nucleotides? These biologically important nucleotides, it can be studied under three categories: the coenzymatic nucleotides, the high energy nucleotides, and the cyclic nucleotides. On here, the coenzymatic nucleotides can be divided further into two based upon the chemical composition: the pyridine nucleotide and the flavin nucleotide. So, what are coenzymes, first of all? When we speak about the enzymes, most of the enzymes, they are referred as apoenzyme. I mean, sorry, what you call holoenzyme. Most of the enzymes are referred as holoenzymes because they are formed of two constituents. One is a protein part, which is known as the apoenzyme, and a non-protein part, which is referred as a coenzyme. Okay, non-protein organic part, it is the coenzyme. So, an apoenzyme and a coenzyme. Apoenzyme, the protein part, and the coenzyme, a non-protein organic molecule. These two together work as an enzyme. Okay, so in that case, we call such an enzyme as a holoenzyme. So only when the coenzyme gets associated with the apoenzyme, it can work as an enzyme. It can catalyze a particular reaction. So those coenzymatic nucleotides are the ones we are referring over here. Okay, the major coenzymatic nucleotides are pyridine nucleotide and flavin nucleotides. Okay, uh, uh, these are actually vitaminous coenzymes. The reason is pyridine, it is what we are referring here, it is the nicotinamide or niacin, it is a vitamin B complex vitamin, and flavin, it originates from riboflavin, yet another vitamin B complex vitamin. Okay, so here we have the pyridine nucleotide and the flavin nucleotides. The major pyridine nucleotides are the NAD or the nicotinamide adenine dinucleotide, and the NADP, the nicotinamide adenine dinucleotide phosphate. While the flavin nucleotide includes FAD and FMN, where FAD is flavin adenine dinucleotide and FMN is flavin mononucleotide. Okay, that is the two flavin nucleotides, which are coenzymatic nucleotides, important in biological systems. The high energy nucleotides are usually high energy nucleotide phosphates, and among that, the most important one is the ATP, that is adenosine triphosphate. Okay, the cyclic nucleotides, they are actually derivatives of purine. So you can see here it is ANG. Okay, the cyclized forms of purine nucleotides, purine mononucleotides, are the ones we refer under cyclic nucleotides, and here it is cyclic AMP and cyclic GMP. We will be studying only the structure of cyclic AMP, which is actually cyclic adenosine monophosphate. Okay, so these are the different categories of biologically important nucleotides, repeating: coenzymatic nucleotides, high energy nucleotides, and cyclic nucleotides. So in this presentation, we'll be looking into only the pyridine nucleotides. Okay, the NAD and the NADP. So what is NAD and how is this they are formed? NAD, or the nicotinamide adenine dinucleotide, it is otherwise known as niacinamide adenine dinucleotide, because the nicotinamide, it is otherwise referred as niacin. When you learn about the vitamins, you will come to know niacin and the nicotinamide, it is niacinamide and nicotinamide, it is just one and the same. Now, it was, and the NAD was the first coenzyme to be identified. Even the structure elucidation was also done later. And this NAD, it serves as a major carrier of electrons or hydrogen in the oxidation of certain substrates. Okay, so it is involved in oxidation and reduction reactions. Such reactions are known as redox reactions. Okay, so NAD, it is involved in oxidation of certain substrates which are in reduced form, or it is involved in the reducing of certain oxidized substrates. And so in both instances, it can play a very important role. Okay, it carries electrons from one reaction to the next. So it takes up a pair of electrons from one reaction and get itself reduced, right? it takes up electrons and it reduces itself, and then it gives these electrons to some other substrates during another redox reaction. So it has a role almost similar to that of ATP. What does ATP do? ATP, it acts as a phosphate carrier. Okay, so it gives off phosphate to some substrates and uh, gets converted into ADP, right? And in some other reactions, what it does, it takes up phosphate from some phosphorylated substrate and becomes ATP. So it is a phosphate carrier. Just like that, NAD, it is considered to be an electron carrier. Okay, NAD accepts electron from a donor substrate, electron donor substrate, gets reduced to NADH, right? And this NADH, it gets oxidized by donating a electron to an electron acceptor, and thus it gets oxidized. So this is how it is. We will see to it. Okay, now NAD, it is formed of two moieties. Okay, one is uh, nicotinamide. Okay, and then we have two molecules of ribose, one molecule of adenine, and two molecules of phosphate, H3PO4. So all these comes together form what is known as a NAD. Okay, the whole structure, it can be divided into two as. Okay, so this part, okay, the first part we can say, which is composed of nicotinamide, then ribose and a phosphate, it is considered to be nicotinamide nucleotide. Okay, it is nicotinamide nucleotide. I repeat, nicotinamide, ribose and phosphate together, it forms a nicotinamide nucleotide. Okay, this is considered to be a pseudonucleotide. Now, what is pseudonucleotide? Here, what happens is the bond between the what you call the nicotinamide and the ribose is not a glycosidic bond. This is not a glycosidic bond. Okay, unlike over here, this is a glycosidic bond between adenine and ribose, isn't it? But here it is not a glycosidic bond. Okay, and nicotinamide is not a purine or a pyrimidine. It is a pyridine derivative. This is a pyridine structure. It is a pyridine derivative, right? So altogether, it makes up a pseudonucleotide. Okay, so this is a nicotinamide nucleotide. This is you can see this one is adenine nucleotide. Adenine, what you call the ribose and the phosphate. So this is adenine nucleotide. So together we have nicotinamide nucleotide and adenine nucleotide bounded across the phosphate bond. Okay, so this makes up nicotinamide adenine dinucleotide. I suppose you got it right. Okay, now the reactions it catalyzes. So here you can see this represents nicotinamide adenine nucleotide. Okay, this is a nicotinamide ribose phosphate bound to the adenine nucleotide, that is adenine, ribose and phosphate. Okay, so this is NAD. Okay, why does plus charge over here? Okay, because it is in oxidized form. NAD is in oxidized form with a positive charge on the nitrogen. Okay, now when it undergoes what you call reduction, what happens is it receives a hydride ion and an electron pair. Okay, this is a hydride ion electron pair, and this is received by the nicotinamide ring. Okay, it is not received by any of the other molecules. It is received on the nicotinamide ring. Okay, so it receives it. What happens? This hydrogen, it gets bound to the nicotinamide ring and it neutralizes the positive charge on the nitrogen. So this is what the product will be. Okay, this one, it is reduced nicotinamide adenine nucleotide, which is known as NADH, which you are familiar with. Okay, so NAD is an oxidized form. When it takes up an electron pair or a hydrogen, it becomes NADH, which is the reduced form. Okay, during this time, okay, this is the overall equation which shows the reaction. Here you can see this is a substrate and the substrate contains hydrogen molecule. Okay, this substrate is in reduced state because it contains a hydrogen. Okay, now this, it is adding up to reacting with NAD plus or the nicotinamide adenine nucleotide. This is oxidized state, right? So when this reaction takes place, what happens? This substrate, which is in reduced form, it undergoes oxidation. It releases the hydrogen. It becomes oxidized, right? What happens to the hydrogen released? It is taken up by the NAD. Okay, and this NAD, which was in the oxidized state, it will become reduced. Okay, so what is happening? A reduced substrate, it becomes oxidized, and an oxidized substrate, it becomes reduced during this reaction. And this is what is known as a redox reaction. So NAD, it is involved in redox reactions. This is one typical example. right? Where pyruvate, it gets converted into lactate with the help of the enzyme lactate dehydrogenase, and it is a redox reaction. Fine. Okay, thank you.